Welcome back. Today we're going to be looking at NCBI, the National Center for Biotechnology Information. This is run by branches of the U.S. government, so it's updated regularly and is free for anyone that has access to the internet. It's a very handy website that's been around for several decades, and it's very handy because of the amount of information within its databases and also the analytic tools you can use on those data sets. So let's take a look. Once we go to ncbi.com, it actually redirects us here. Once we get here, we can do the drop-down menu to look at all the different databases available to us. Now, some of the more popular ones can serve domain. This will show you domains within a protein. Gene. This gives you the raw sequence for genes. Genome. This will give you the genome for over 100,000 different organisms, from archaea to bacteria to eukaryotes. The gene expression omnibus. This is a great source for microarray data. Taking a look further down the list, nucleotide. Nucleotide is a great database for getting DNA sequences. OMIM, the Online Mendelian Inheritance in Man database. This is a non-sequence-based database that correlates genes with particular diseases in humans. It's a great starting place if you want to take a look at what genes are associated with what disease. Protein. Here's where you get protein sequences. And of course, everyone's favorite, Pubmed, which is the overall encompassing database repository for medical and scientific literature. This should have been discussed to you in prior classes. So let's take a look at how you get some of the data. Okay. We can type in general terms. So let's say I want acetyl lactate, which is an enzyme responsible for making branched chain amino acids. I didn't specify what organism I wanted it from, so I have several different DNA and RNA sequences, actually about over 400,000 of them. I have almost 13,000 different genes that match that description, and I also have quite a few proteins. So depending on what I wanted to look for, I could click on any of these. Let's go ahead and click gene for now. From here, it'll start to give me a list with gene symbol, description with whatever organism it came from, its location on the chromosome, and then any other aliases that that gene might have. Now, if I'm looking for this particular protein or gene within a particular species, I can either type it in here, and it will limit my search that way. Or if you notice on the right-hand side, I have top organisms. So I could take a look at only the ones from Staphylococcus aureus, for example, by clicking on it. There it goes, okay? And so these are all the hits for acetolactate in Staphylococcus aureus. So you get the idea there. Let's say this first one, or actually we'll go with the third one, this acetolactate synthase. This is the one I'm interested in for whatever particular reason. So we can go ahead and click on that, and it will give us more information, what strain it might have come from, the lineage of the organism, if there are any other synonyms that have been used in past databases, etc. Okay, you can even check out domains within the genes, etc. down here. But what's really interesting when we do that is what's called the Fasta. So if you click on that, this gives you the raw sequence. And you'll notice you have your start codon. You'll notice you have a stop codon. So we know this is a gene that would encode a protein. We can then do analyses with this, either within NCBI or in other programs. So quite a few algorithms will recognize Fasta sequence from this caret and then recognize the DNA associated with that. Okay. If I want to, for example, do sequence alignment, I could do run blast, etc., etc. Okay. I can also take a look at the protein sequence for this particular gene. Okay. It looks like we're getting a bunch of hypotheticals here, so actually let's go protein. We will type in acetolactate. Okay. And oh, we like Staphylococcus, so we'll just do staph again. Give me staph. Well, I guess they are that small. Okay, fine. So regulatory subunits. Do I have a whole protein anywhere in here? No. Catalytic. Yeah, I'm going to go with a catalytic. Okay. So here I have a catalytic subunit from my acetolactate protein. I can click on it to get more information, just like I could for the gene variation that we saw. Okay, so it's telling me my organism, it's giving me any publication data associated with it. Here's a link to the Pubmed article if I wanted to read it. I can scroll down and take a look at the different domains, etc., that are associated with it. Here's my sequence, but notice it's not in Fasta format, so if I want to do that, I come up here and click Fasta. And lo and behold, there's my beloved carrot that then gives me my protein sequence. And similarly, I can do sequence alignment using BLAST. I can identify conserved domains, and so we will visit these areas in another lecture. The other thing I'd like to show you while I'm here is taking a look at genome. So let's say, for example, I want the genome of a particular species. Or if I'm still dealing with my Staphylococcus example, Staphylococcus has several different strains. Some of them are pathogenic. So I can type in whatever organism I'm looking for, go ahead and find it in my list and click on it. And you'll notice that I've got one chromosome in this organism. There's 4 plasmids that have been sequenced from this organism. It'll give me some statistics about the length, the protein count, percent GC, publications, etc., etc. I can also come down here and take a look at protein table. So I'll do that in just a second. And I can also look at the raw data for that genome. Okay. So notice the whole genome is going to be rather large. It might take some time to download, so hopefully you've got a decent internet connection. Okay, and of course here's every single gene as we go through the genome. It's locust ID tags. You can click on it for more information, etc., etc. But if I scroll all the way down to the bottom, notice I no longer have my sequence there, so again I've got to go to Fasta. And it's going to download literally thousands upon millions of A, T, C's, and G's for whatever analysis that you deem worthy. Okay. Now remember when we clicked on the protein table right here, we get this kind of a view, which is very helpful. So here I have all the proteins in one large table that are associated with that particular organism. I can download the table. It gives me where it's located on the chromosome, if it's the positive or negative strand of DNA, whatever gene ID, its gene symbol. This will then give you protein product, which is your protein sequence, length, protein names, etc. This is completely searchable, so if I want to type in something like DNA polymerase, it's going to give me all the different DNA polymerases that it found within that genome. Okay. With that said, then I can take a look at different strains, and that is also very helpful. So if I go back to my genome of my original organism, Staphylococcus aureus, and now I'm going to take a look at the strain, so I want to browse the list. You'll notice there are several different strains of Staphylococcus aureus. That kind of makes sense given the number of patient isolates, considering this is a highly resistant organism. You'll get an idea then of the level of completeness of a particular genome. So if I'm almost all black, that's complete. Some will just have chromosomes, some will just have scaffold or contigs, which are partial sequencing. So keep that in mind. You want to hopefully look for the ones that are the most complete. It'll tell you how many genes they found, how many proteins, when it was released, if it's been modified. If you want to FTP download it, that's no problem. That's right there. If there are any plasmids that were associated with that particular strain, we can click on them and get their data as well. Okay. So it's very handy if you want to do strain-strain comparisons or anything of that type. Well, this has been a short tutorial of NCBI and its entrees search factor, and you've been introduced to Fasta. So I look forward to having you guys search out sequences for homework. Let me know if you have any questions, and enjoy.